Of course. So I'd like to add a quick warning. I do have two kids and it's getting close to bedtime, so it may get a little noisy at some points and they may bust in here, but I'll try and do my best around that. So just bear with me. So thank you everyone for being here today. I'm excited to talk about fungal DNA barcoding. It's really important and it's come a long way in the past few decades. It's this course, or what I'm going to be talking about today, is mainly going to be theory and kind of the biology behind it, the rationale for why we use the particular region of fungal DNA that we use to identify fungi. And so we're not really going to talk about applied aspects of extraction and PCR and sequencing. We'll touch on them, but basically it's just why we're doing what we're doing. So you'll understand the process, which makes it a lot easier to learn, in my opinion. So if you have any questions, if you want more clarification on anything as I go through, please put it in the chat and Angel will help me get those questions. Okay. So what we'll cover today is, why do we care about fungal DNA? Why can't we just use physical characteristics? That's called morphology. What is DNA extraction? What is DNA amplification, where we make multiple copies of DNA? We'll touch on sequencing. I'm not going to go into a lot of detail. It's really interesting, but it's also really dry, so we're not going to talk about that a lot. Fungal species identification, and then really what defines a species. And then we'll have more time at the end for any questions I'm not able to get to. So what is DNA? DNA is deoxyribonucleic acid, and it's really codes for all information in all living organisms. So DNA is made up of two backbones that connect bases with hydrogen bonds, and so it makes this double helix. And then there are only 4 bases that make up all of our DNA. So humans have about 3 billion bases in our genome. So it's really long. And really, it's just made up of these 4 bases. And so you have a set connections. Cytosine always pairs with guanine on either side, and then adenine with thymine. And so it makes it really easy. If you know one sequence of DNA, you can figure out the other side and you can make multiple copies of it. And then the uniqueness of it makes it easy, or we're able to use it to identify species. So more closely related things have more similar DNA. So us and apes have more similar DNA than us and a bat or a dinosaur would. But we all come from a common ancestor. So we use fungal DNA to help with species identification. This is especially important because some fungi, as I'm sure all of you have experience with, are hard to identify using just macroscopic characteristics. So that's what you're able to see with your visible eye. Some of them are really easy to do. If you see chicken of the woods, it's very hard to mess up once you learn the characteristics you're looking for. There's not a lot of other things that look like it in your area. But there's other things that are really hard to identify. You have to go into microscopic characteristics, and even with that, with the help of a microscope, you sometimes can't tell them apart. So only about 1% of fungi make mushrooms. And there's estimated to be, estimates range widely, but we're going to say about 4 million species of fungi. So that's a lot that don't make mushrooms. And they just make hyphae, or there are yeast single-celled fungi. Another complication is that when you grow things on a plate in culture, or even if they mushroom, they can change how they look based on the environment. So what they're growing with, whether it's dry or wet, different types of nutrients it grows on, it can change how it looks, its morphology. So having other ways to identify species is important, and DNA is something that does not change, or at least not quickly. So we're able to use it to identify species. DNA is also really important to help figure out fungal diversity. So if you have a survey that you're conducting in an area, if you go out, collect, this was collected in one day in Florida, I guess 2 summers ago now, and it's pretty diverse for about an hour and a half, although there were about 40 mycologists out there looking. So we were able to collect a lot. But if you wanted to identify all of these species, it would take some time. If you're familiar with the area and the species, then you're probably able to knock out quite a few, and you only have to dig in to the details, microscopic features of a few. But if you're new, or if you're just trying to survey an area that's never been surveyed before, being able to just do an extraction, pull out the DNA, and quickly identify it is really useful. And then that can get deposited into a database along with the specimen to be used forever after by other mycologists. Another thing which we'll talk a little bit about later on is called environmental sequencing. So that's kind of came about in maybe the 2010s, where you can take a sample of soil or a sample of water or air, and you're able to extract all the DNA that's in it. So 1 gram of soil can have hundreds of fungal species in it. Some of them are active, some of them are dormant in spores, some of them are dead, so you just have like remnant DNA left. But you're able to extract all the DNA in it and sequence all of it. So you can find out all the species that are in a sample of soil or water, and you can kind of get an idea of what the diversity is. You can kind of figure out a little bit what could they be doing in that soil sample. Okay, and so it's really important for fungal ecology. If you're able to only look at mushrooms to figure out what species are there, you're really limited by what's able to fruit at that time or during the window where you're observing. If I was doing a study looking at what fungi grow in the juniper forest here, I would have to do multiple surveys over a very long time to capture what species are fruiting. And that wouldn't be everything, because not everything fruits. So it's really helpful for fungal ecology and really has advanced our understanding of fungi. And then also conservation, which thankfully fungi are really being recognized for their importance, and they're being conserved more and more. Just since the past 10 years, fungi have been recognized and put on these federally protected lists, and you can see it's increasing. So having DNA is really important because you have people who can do surveys that don't have to be specialists. They don't have to be mycologists. They can just go out, do surveys, figure out if this area that's being developed has this really rare fungus on it that needs to be protected using DNA, and then that land can be protected. Okay, hang on, having some issues here. There we go. Okay. So DNA extraction, how do we get at the DNA? DNA is within the nucleus inside cells, and you have to kind of break open the cell to access the DNA. If you just try and copy DNA without opening the cell, it's not really going to work. So this picture here is a fungal hyphae. So you have the growing tip here called the Spitzenkorper, which is in red. And then all of these greens in this confocal microscopy picture are nuclei. So this is all DNA, little packets of DNA. And so you're trying to access that. So you need to break open the cell. And there's 3 main ways that you do this. Mechanical, which can be with a mortar and pestle. You can use, there's these teeny tiny beads that come in many different sizes and shapes and types of material that you can put into a 1.5 milliliter tube, and you just put it into machine and it beats it. It's the technical term for it, and it lyses the cells, so it breaks them open. Additionally, you can use heat, and then there's chemical ways to lyse the cells. Most methods, I think, use all 3 together. I use a really simple method for extraction. It takes like 10 minutes and uses 2 different solutions, but it uses all 3. And that's the best way to do it. And then the extracted DNA is stored in a buffer, which keeps the DNA stable. Double-stranded DNA is pretty stable. It's unlikely to break down if you leave it out at room temperature. I've done that before in grad school, left DNA out overnight, and it was totally fine. RNA, though, which is single-stranded DNA, so instead of having the double helix, you just have one side. that is less stable. So DNA is pretty stable. Working with it is much easier than working with RNA. So when you break open a cell, you don't just get out DNA, you get out a ton of other things. So here, this is a kind of cartoon of what else is in a cell. This is of corn smut, Ustulago matus. And so you have many other things. Here you have the nucleus with the nuclei in it, but all these other things come out as well. So a lot of extraction techniques will have filtering steps too, because a lot of these things can kind of inhibit your replication of the DNA, which we'll talk about in a minute. So that's just something I want you to be aware of. Not all extraction kits use this, or extraction methods require it. But some species of DNA or species of fungi produce chemicals that you have to remove these chemicals before you're able to amplify the DNA. Okay. So once we extract the DNA, we're going to amplify it. So that's just making many copies of a particular area in your target DNA. And we'll talk more about the particular area in a minute. Right now, we're just going to talk about the process of replication. So to do this, scientists have, not me, but really intelligent scientists have figured out a way to kind of co-opt the natural process of DNA replication. So just to remind you, we start out with this DNA double helix, where we have our nucleotides, our 4 nucleotides, bonded together by hydrogen bonds. So the first thing that we have to do is break these hydrogen bonds and open up the helix. And then we're able to replicate each strand. So how is this achieved in the cell? There's a number of different kind of machines that operate in the cell that do this, and they all work simultaneously within your cell to make new copies of DNA. First thing, you have something called helicase, which unzips your DNA, breaks these hydrogen bonds. Then you have these primer sites. So to copy the DNA, you have to have a spot where DNA polymerase can attach. It has to have a starting spot. If you think of like a zipper on your jacket, you have to first connect it at the bottom of your jacket and then zip up to close it. And so it's kind of like that. This is kind of like the starting spot where you start zipping. And then DNA polymerase, which actually copies the DNA. And it does that by reading what's on this side, these bases here, and it takes these dNTPs, which are just the nucleotides that we talked about, those 4 nucleotides, and it adds them on. So if there's an A, it adds a T. If there's a C, it adds a G, and so on and so forth. And so this is all that PCR, polymerase chain reaction, does. You first unzip the double-stranded DNA, break the hydrogen bonds, and this is a step called denaturation. Then you add your start site or your primers to this unzipped DNA, and this is called annealing. And then the DNA polymerase will add these bases, dNTPs, to the starting at the primers and create new copies. And this is just extension. And so the polymerase chain reaction, you add in all of these components, your DNA, your primers, your dNTPs, which are the nucleotides, and your DNA polymerase. And you just make a mix of all this together and you put it in your machine. And each of these steps happens at a different temperature. And it just repeats this over and over and over again. And then you exponentially increase the amount of DNA you have. Say you start with just 1 copy of DNA, which usually you'll start with more. It's hard to isolate just 1 cell of DNA of fungi. And then 1 turns into 2, 2 turns into 4, and so on and so forth. So you end up with thousands to millions of copies of your DNA. Usually, when I run a thermocycler program, I run it for 30 cycles. So you end up with a lot of DNA. And so this isn't all the DNA in fungal cells. It's just that one region that you are amplifying. So after you have your DNA, after you amplify it on the thermocycler, run PCR, you have to sequence it. So old thermocyclers used to look like this. And so you would have these 3 water baths, and they would be at 3 different temperatures. And so each bath would be doing one of these steps. So this first one would be at one temperature, and this would be doing denaturation, so unzipping the DNA. Second one would be annealing of the primers to the DNA, and the third would be extension, so making the copies. And so previously, before my time, this was like my PI's time, you would have to stand here and you would move your samples between each bath over and over and over again, like for 30 cycles. And so each step is maybe like a minute. So you'd be standing here for, an hour or two, depending on how fast your DNA polymerase makes copies. So not much fun. So when a thermocycler was introduced, where it does it automatically, it made things a lot easier and a lot faster. And so this type of PCR is commonly used for Sanger sequencing, which is really the first generation of sequencing. If you hear people talk about like first generation, next generation, this is the first generation of sequencing. So when you do Sanger sequencing, you end up with these chromatograms. And what this is, if you notice, there's 4 different colors, and that corresponds to each of the bases. So when it's being sequenced using Sanger sequencing, it uses these fluorescent dyes that flash a color when a different base is detected. And then the computer reads it, and it puts out these really nice chromatograms that tell you what base flashed. And so you get your sequence. So with this first generation of sequencing, Sanger sequencing, you get fairly short regions of DNA. For fungal ITS, I normally get around 1,000 to 1,200 base pairs. So those are, A's, C's, T's, and G's, 1,000 to 1,200 of them. And the cost is pretty expensive. So for 1 billion bases, which remember, the human genome is about 3 billion bases, roughly. It's $2.4 million per 1 billion base. So for a human genome, if I was to use this first generation of sequencing, it would be about $7 million. Pretty expensive. And then a limitation of it is that you need pure DNA. So if you want to figure out what what species a mushroom is, you have to culture it onto agar, get it into pure culture so nothing else is growing but that one fungus that you're interested in, and then do the extraction. If you don't, you end up with multiple peaks because it's amplifying multiple different kinds of DNA, and so you don't get good what's called resolution, and it's a mess. You can't figure out anything from it. So it's pretty sensitive and it's very expensive. Okay, so that brought us to the second generation, what was previously called next generation sequencing, which is now older technology. And so this really, the aim of it was to make it sequencing happen more quickly and also much more cheaper. So with this, you were able to sequence multiple regions in parallel. So what that means, instead of having one pure culture on a plate that you extract and sequence, you're able to kind of just take a soil sample and sequence it and get everything in it at the same time. So instead of running multiple sequencing runs, you just run it all together. So this is really important for whole genome sequencing. You could take a whole bunch of DNA from one organism, and you could cut it up into a bunch of different sizes, select for what size of DNA you wanted, so how many base pairs, and then sequence it. And that way you can kind of piece together a full genome. And then also this microbiome characterization, which I talked about with soil, or taking some fecal matter and finding out what a human's microbiome is. Has it changed because they're sick or whatever? And so with this though, here I'm showing Illumina MySeq. And so for Illumina, you can get about 250 to 300 base pairs. So pretty short. And if you're doing microbiome characterization, you're sequencing this one region of interest, which we'll talk about. But for each species, you're only getting about 250 to 300 base pairs, which is really short. So you can think, if you're trying to identify a species, having more bases is a lot better. Trying to think of an analogy I could use. Okay, if you're doing a puzzle, 500-piece puzzle and you have 10 pieces, it's kind of hard to figure out what the puzzle is going to be. The more pieces you have, the easier it is to figure out what the puzzle is going to be. So it's kind of like that. But the cost is astronomically lower. So for 1 billion bases, it's $5 to $150. So if you remember, Sanger sequencing, the first generation was millions of dollars, $2.4 million for 1 billion bases. So it comes way down. It's a lot faster and a lot cheaper. So the third generation is this nanoion PacBio group, which is pretty amazing because you're able to get much longer reads. It's still cheap, but you can average about 10 to 20,000 bases, which is huge. And then I was reading somewhere that Oxford Nanopore, the longest read so far that they've been able to get is about 4 million bases, which is huge. That's like full human genome. So pretty crazy. And then it's also cheap. So it's really good. And then you have these ones that are personal. So like this mini ion, which is crazy and I'm really excited about. It costs about $1,000 to get the starter pack to do at home. And you can just plug it into your computer and sequence stuff at home on your computer. I really want to get one, but I'm trying to hold off for a bit. So much more accessible to people. Okay, so we extracted our DNA, we amplified it, and we sequenced it. So now what do we do with it? So DNA has regions that code for proteins, and among other things. And so these coding regions are called genic regions, and the regions in between are called intergenic regions. So the coding regions are really important because they code for important things that we need, proteins. And these intergenic regions don't code really for anything, so they're not as important. And so you can think that these coding regions have slower rates of mutation in these intergenic regions. So here, mutation, I mean, at the smallest level, changes in one base. So it changes from a C to a T. So if you have like this protein that's, I don't know, let's estimate 1,000 bases long, you have a mutation that changes one single base in it. When we're replicating DNA, we have machinery that'll come in and it'll find that change, that mutation, and it'll fix it. Because any changes in that protein and that genic region can change the protein, which could be really bad. It could kill us. And so it's really important that we have these things that fix these errors that can arise. These intergenic regions, though, when they have mutations arise in them, it doesn't really matter because they don't code for anything. So they can accumulate mutations at a faster rate. So you have different rates of evolution. So like I said, more closely related individuals have higher percent of DNA in common than the ones that are more distantly related. So we can kind of use these regions with differing rates of evolution of mutation and use them to help us identify species. But you have to find the right area, and that's the hard thing. And this is based on a pretty big assumption, which for most cases is safe, but sometimes isn't. And that's variation between species. So between us and dogs is greater than what we would find between myself and my sister or myself and my children. So within species, which most of the time is the case, but you know, there's always exceptions. There's no absolutes in science. So for fungi, it was determined in the early 2000s that we should use ribosomal DNA region. Okay, so this is a really highly conserved region because it's very important. So it codes for what's called a ribosome, which if you remember from way back, old biology, biology 1, ribosomes make proteins. So it's actually kind of cool because ribosomes aren't proteins themselves. They're mRNA, so that's single-stranded DNA that's folded, and then it takes other RNA and it reads it and makes proteins. So it's really important because if you can't translate what's in your DNA into the end product, you won't exist anymore. So these have very low rates of mutation. So they're highly conserved. And so what we're seeing here is the region. So these black regions are the genic, the coding regions that are more highly conserved. And then these thinner spaces in between, this is called the intertranscribed spacer region, are the intergenic regions. So these are the areas that don't code for anything. So you can expect faster rates of mutation in these areas than in these areas. Okay. So we can kind of use this. So for ITS1 and ITS2, these intergenic regions, these are really good for species level resolution. So if I want to determine whether these two mushrooms, which look exactly the same, are the same species or not, I would use this region. Excuse me. But if I want to look at higher level classification, so maybe like family, I could look at these coding regions, which have slower rates of evolution. So if I were to use this region right here, or this region right here, to look at species level resolution, so if I want to determine whether these two fungi are different species, I wouldn't be able to, because there wouldn't be enough mutations difference in the base pairs with these regions. I have to use these regions. But if I want to look at, are these different families, then I could use this region. So just there's a lot of variation in this area that's really good if you want to figure out whether things are the same or different. Okay, so I took some DNA and I sequenced it and I got some sequences back. So these are the final sequences that I got back. And it's really small here. I should have gone in to make this bigger, so I apologize. But here I have 4 species of Ladyporus. So that's chicken of the woods genus. This first one is chicken of the woods. And then there's other ones in the same genus, but different species. And then I have some other genera of fungi that are more distantly related. So I have a Trametes, Tuberia, and a Boletus. So you can see that these first 4 are pretty similar. There are no differences in this region. Oh, except for this one right here. You can see that these 3 right here have a T, and this one has a C. So slight difference. So you could suspect that this one is the Ladyporus carabensis is slightly more distantly related or has mutated a bit more. But if you look at these next 3 that are not of the same genus, you see a lot more differences. This one right here, this Boletus, has an insertion. So you can see that these other species don't have this nucleotide right here. So this one, there was a mutation where it just inserted a T. And then if you go along, you see other differences. These 3 species have a T here versus the C here. So you can kind of go along and compare these sequences that you get back and look at how many differences there are. The more differences there are in the bases, and if there's insertions or maybe a nucleotide is completely missing, so there's a deletion, you can kind of figure out how distantly related it is. Excuse me. So this is sequence similarity. And so the question here becomes, what's the cutoff? Do you need complete similarity for it to be the same species, or can you have like a few bases be different and it be considered the same species? And this is where it gets a little hard to determine. It kind of depends on what group you're looking at. So for ectomycorrhizal fungi, generally we use 97% sequence similarity. So for every 100 base pairs, there can only be about 3 that are different. If there's more than that, then you would consider it a different species. For endophytic fungi, we normally use 95%, and that really comes down to taxonomy. So most ectomycorrhizal fungi, not all, but most are part of Basidiomycota. And then most, but not all, endophytes are part of Ascomycota. So you have different rates of mutations based on what taxonomic group you are. So this is something that you can do if you want to. You can look if you have a new species that you're trying to define, if you're trying to determine if it is a new species or is it the same as another, you can compare the sequences and try and figure out whether it's different or not. Another thing that you can do is you can just take your sequence that you get and put it into NCBI, which is a online database of sequences, and then you do what's called blasting it, where it compares it to all the other sequences in the entire database, and it will tell you what the closest ones are to it. So you can just quickly find out what species it is. But this is what it's doing. It's taking all of the sequences it has and it's comparing them. Unfortunately, there are some issues with this. There are groups with really poor species level differences in the ITS region. So a lot of the Ascomycotis, so Cladosporium, Aspergillus, Fusarium, Penicillium, you can't use this region that's been defined as the region for fungi to look at species level. What you end up with is all of them just come up as Aspergillus or Fusarium or Cladosporium. And it's called a species complex, where you're not able to figure out which ones are actually different species. Even though looking at them, they could look completely different. One could be found in South Africa and another one could be found in Norway. So they don't live close to each other. One could live at the top of a mountain, another could live in a tropical forest. So they don't really have the same ecological niche. So it doesn't seem like they'd be the same species, but you aren't able to tell from this region. So you would have to sequence a completely different region of DNA to figure out whether they're different species or not. So this brings us to my favorite part, is what defines a species. So historically, I think the most common method that probably all of us have used before is a morphological species concept. So this is based on morphological or physical differences. And it's based on the assumption that things that look more different are more distantly related. So for some organisms, that works really well. So for us and apes, you can tell that we are distantly related, although we are related. But for fungi, it's a little difficult. So I'm probably gonna get this wrong, but I think like chimps and us have 3% DNA difference, I believe. But if you were to look at two fungi that only have 3% difference in DNA, they could look pretty similar. So when you're looking at these, kind of cryptic species that don't produce mushrooms, and some of them don't have any sexual states that have ever been observed. Some of them only produce asexually. It's hard to discern these morphological differences. So Lentinus tigrinus is a good example of this. So it produces this, I think it's called gasteroid morphology, where you have the gills completely enclosed. So this doesn't happen all the time. You will have gills that are open and exposed, but sometimes it produces this type that's completely covered. It just has a layer of like hyphae over it. And so for a long time, this was classified as a different species, even in a different genus, based on morphological characteristics, which, I mean, I would have classified it completely differently. But they figured out that they could actually mate this kind of weird, funky gasteroid species with the one with gills. And so it seemed like it was the same species. So this brings us to the biological species concept, which is species are defined as an interbreeding population. So if they're able to reproduce and create offspring, then they are the same species. But you run into issues with that, even outside of fungi. So if you think of like a liger, it's a tiger and a lion, they can mate and create offspring. I'm pretty sure it's sterile, but they still produce offspring. But they're obviously different species. They look completely different. They're found in different places. So it's hard with fungi because, again, not all of them reproduce sexually. Some of them only reproduce asexually, and some don't even do that. So biological species concept is a little difficult to use for fungi. They like to be difficult. So another option is the ecological species concept. So this is based on ecological niche. So this is the idea that if things kind of have different modes of living. So then they won't really interbreed, and their DNA will gradually separate over time. So here we have two species of chanterelles that look fairly similar. This one, though, over on the left is found on beaches and sandy soil, and I think is mycorrhizal with sea grape. I don't know. I can't remember. It's mycorrhizal with one species of plant. And then the one on the right is found in hardwood forest. And it's mycorrhizal with a number of different hosts, like beech and I think oak. So even though they look kind of similar and might have been classified as the same species based on morphology, they have different ecological modes. And then the last species concept that we'll talk about, there are others, is the phylogenetic species concept. And this is generally based on DNA. So it's kind of taking these, physical differences that can arise due to separations of DNA, this inability to interbreed due to differences in DNA, and then ecological differences, and just bringing it down to DNA, so the most basic level. So it's thought that, with time, you'll have separation of genetic code, you'll have more mutations that are different between them, and so they'll separate into different species. And as I've mentioned, there's already issues with that based on what region you use. With some of them, you can't really tell a difference. And so, what defines a species? You really have to use all of these. You have to use all of these tools that are at our disposal. You use morphology, both macromorphology and micromorphology, which is one of the cooler things to look at. Looking at spores under a microscope is lots of fun. Look at the ability to interbreed if you are able to do crosses. That it's hard to do with fungi. Consider the ecology. So do they have the same host? Do they cause the same kind of effect on a different host? So if you think of like a pathogen, there's some endophytes that will cause disease on some host, and then they'll be beneficial to other hosts. And so if you have one species that, based on the ITS region, they seem like the same species, but they have a different effect on the host. One causes disease and one doesn't, which there are cases of this, then they're probably different species. And then genetic differences. So using all of these tools at our disposal, we can figure out what a species is. But my favorite thing to consider is like, when does a new species arise? And over time, and it's fun to think about, but. Okay, so that concludes my talk. If you have any questions, please let me know. I think that didn't take too long, so we have plenty of time.
Yeah, so Lisa White did have a question about how long is the amplicon that is sequenced?
Uh, for fungi,
Yeah.
so generally, it depends on what primers you use. So let me go back to the picture of this region. Okay. So you can design different primers. So if we look at here, so primers are designed, the ones that we use in PCR are designed by us. So you can make any primer you want to, and they're actually really cheap, which is nice. So you can just gather some sequences from NCBI of what you want to look at, and you can look and see, okay, so I have this region here that's all the same within this latyporous group. So I can make a primer that starts at this G, and it's GAATC, so on and so forth, all the way down to the end. And that way, when I sequence and put that primer in, it will amplify this one region that I know is unique to latyporous. So you can design it however you want to, so you can amplify as much of a region as you want to, What really is your limitation, though, is what sequencing technology you're using. So I use Sanger and Illumina mostly. So for Sanger sequencing, the primers I use, the forward primer is here and the reverse primer is here. So it'll sequence forward and reverse, giving me a double-stranded DNA, and that's about 1,200 base pairs. When I do Illumina, though, you can't sequence 1,200 base pairs. You can only get about 250 to 300 if you're lucky. So I usually use the ITS2 or the ITS1 region, and I get about 250 base pairs. So it depends. And then if you use the nanopore, you can get 4 million bases, I think it was. So depends on what you're looking for. But if you're just doing, this ITS region, it's about 1,200 bases.
Okay. And Helena asks, how often do you find a new species?
So I've never been lucky enough to find a new species, but that's a hard question to answer. So with environmental sequencing, we've had the kind of like this explosion of how many species we think are out there. Um, but the issue is when we take a soil sample and sequence all the DNA in it using Illumina, we end up with only 250 to 300 base pairs of that one species, which isn't really enough to describe a new species. So to actually get to name a species, and you have to formally describe it. So you have to publish a paper where you describe the morphology of it, you talk about the ecology of it, you sequence it, you have to do all of these things. And if you just have 250 base pairs of DNA, but you don't have the actual fungus, you can't really describe new species. So I might have found a whole bunch of new species, but I can't really describe them. So it's a hard question to answer.
Yeah. And I can add to that too, just kind of following what like Stephen Russell from the Hoosier Mycological Society and what Kyle from Ohio DNA, what they're working on. They're doing a lot of nanopore sequencing,
Mm,
and they're going to like the NAMA forays and a lot of these big forays throughout the country. And they also do the North American Myco Blitz, which happens um, different seasons all year round, where people can send in specimens and they do the DNA sequencing. From what I've been told, they're finding a lot of new species,
awesome.
but there's not like it's happening so rapidly, like going into the DNA sort of pile to get sequenced that they need more people that can do the describing of these mushrooms because there's not enough data being collected at the same time that's talking about the habitat, where it was found, the ecology, all of these morphology, these sort of things. Like people that are going out and collecting aren't necessarily trained to do field work as well. So that's the challenge they're running into. There's all these new sort of species being named that are just given these generic numbers because there's no one in academia to do this descriptive work.
Yeah,
And so that's kind of the challenge that I'm seeing kind of happening in the fungal taxonomy world right now.
yeah.
Uh,
Unfortunately, taxonomy's kind of fallen by the wayside, and there are some really good taxonomists out there, but it's not formally taught a lot in school anymore. But like Alan Rockefeller, who is not like a formally trained mycologist, he's a great taxonomist. David
mhm,
Lewis, also not formally trained mycologist, didn't go to school for it. Amazing taxonomist. He's found a number of species in East Texas in his front yard.
mhm,
So Texas is pretty poorly surveyed in terms of fungi. So I know that there are lots of new species that have not been discovered of macrofungi in
mhm.
Texas. So there's definitely space for it.
Yeah. And as you were describing too, just even doing a soil sample, like learning the diversity of that soil and how much there's just undescribed and just
Yeah.
the microscopic level of the soil. Um, that's good to know. So let's see. Some good comments from Rachel. Great job presenting really complicated stuff in a relatively easy to understand.
Oh, good. Thank you for that. It was hard. I was trying to break it down.
And then Candace asks, can the nanopore do microbiome sequencing, aka soil, or do you need to isolate a specific species for that?
So supposedly you can. I've been reading a whole bunch of papers on nanopore and now I mean yeah, I supposedly you can. I don't think there's a lot of studies yet with fungi. I know that there is, oh man, I'm drawing a blank on his name. There's a man on who has a Facebook group of sequencing. Jonathan, I'm sorry, I can't remember his name right now. I'll try and remember it and send it out. But he's teaching a class on mini ion in March that I signed up for, and it's for like doing ITS barcoding. Excuse me. So we're doing species identification, but I'm going to be asking about that. But I don't see any reason why not. I think the limitation there is it's a relatively new um, method, and so there just haven't been a lot of protocols that have been developed for fungi, but I'm sure very quickly there will be because it's so accessible.
Yeah, I'll go do a quick Google and see if I can find that class. Is it in the fungal sequencing Facebook group?
It is, it is.
Okay, I'll look and see if I can find it before the end here. Um, so Keith asks, and I had this question too when you shared the sort of blasts.
Yeah.
Um, how good are the databases for identifying um, what is in your sample?
So that's a very good question and a very good thing to be thinking about too, because your identification is only good as the database it's being compared to. And NCBI does have some issues to it. So because, with these environmental samples, there are so many new species that are being found. Previously, to find a species, you would have to culture it and then describe it. But a lot of things are not culturable. They have to have specific conditions, environments to be cultured. So these environmental sequencing and microbiome sequencing efforts are producing all of these new sequences that are relatively short right now. And you don't have a lot of these in there that are identified. So you can only compare them to what has been identified in there. So you do have a lot of unknowns. So for instance, I did some work in South Texas. I did some sequencing of soil. And I ended up with like 300 to 400 species of fungi, but I was only able to identify down to maybe like order level that I felt comfortable at that level. Um, Maybe like 40 to 50% of them. So yeah, there's a lot of unknowns. And some of them I've identified lower, but they didn't have, you get a confidence score of like how confident it is that this is a close match. And a lot of those confidence scores were not very good. So I didn't feel comfortable labeling them as that. So yeah, that's a very good question. We need better databases, honestly.
And what did you say the name of the database was again?
So it's NCBI. Here, I can pull
Okay.
it up.
And I did find the class that's coming up, and that is Stephen Russell with the Hoosier
That's
Mycological
Stephen
Society.
Russell. Yeah, he does a lot of
Mycoda.
really good work.
So mycoda.com, I believe, or .org. Yeah, mycoda dot, and I'm gonna drop it. There's a class coming up later this I think in March. Um, but if you do submit things while they're doing their Myco Blitz. I know there's one on the West Coast right now, Continental Myco Blitz. It goes from the 12th of January to the 28th. You can send in any sample and they have protocols for preserving them before you send them in, which is simply dehydrating and putting them into a freezer um,
Mm,
before you send, um, which is a
um,
good lead into the next question, which is about how to preserve your specimens for future DNA extraction and sequencing.
that's good. So really, I mean, if you can use specimens like in herbaria, so drying them is good. That's completely fine to do. Um, Otherwise, I guess it depends on what your specimen is. That's a good place to start. So if you have like a mold you want to identify, you could just take like a 1.5 milliliter tube, take a cutting of it and just put it into a tube and maybe freeze it. If it's a mushroom, you'd want to dry it first. But honestly, I think the best thing to do is, having a specimen, a DNA sequence is good, but having the associated data with it. So where you collected it, was it growing on wood? Was it growing on dead wood? Was it growing on the ground? So
Mhm.
what was it growing on? What was around it? Anything like that is really helpful too. So we can get at, what is it doing? What does it do? And having that data is important.
Yes. And I know with Mycota, they at all of the events when they do DNA barcoding or when you do the Myco Blitz, they require a iNaturalist unique ID associated with each specimen, and
That's good.
that's kind of what they use.
Yeah.
So if you don't already use iNaturalist, get on board. It's super fun. It's a great community science project,
Yeah.
and all that data you can put it in the notes. And then the metadata, some of it's already there just based on the EXIF data that's taken from your photo off your camera, like the GPS coordinates. And there's pattern recognition built into it to help kind of get you into a ballpark.
I see a question from Betty/Elizabeth. So it seems like trying to ID all the parts of a diverse sample like soil would involve multiple primers and sequences. Can you briefly describe this process? So generally you just use one set of primers, but that does have issues. So you have something called primer bias, which is something to be aware of. Different primers work better for different regions. And so that's kind of like what I was talking about with those lattiporous ones. The primers that are generally used, so like ITS1F is really popular and ITS4 or LR3, those are really biased toward Basidiomycota and Ascomycota. So if you look at these environmental sequencing papers, you'll find a lot of Ascomycota and Basidiomycota, but you're missing a lot of like the basal fungal groups. So this can be like chytrids, which are definitely present in soil. Previously Zygomycota, which has now been broken up. And even like AM fungi, which are Glomeromycotina, which are, a separate group from Ascomycota and Basidiomycota, they can be less amplified, even though they are there. The primers that you use might not pick them up. So it is better to use multiple primers, 100%. And if you were to do that, what you would have to do is you would have extract your DNA from your soil sample, let's say, and then you would use the same DNA that you have and you would run a PCR of it using different primers. So you'd have to use different PCR mixes with different primers and run it on the thermocycler and amplify those regions that those particular primers are doing. And then you could sequence it on the same run. So the only step you would have to separate is the actual amplification of that region. But a lot of times people don't do this because it's expensive, and academia is a little at a deficit for money, which is one of the issues. So a lot of times these lower groups are completely ignored.
Okay, great. Well, um, let's
Well,
see.
someone asked about the
Me.
graphic background. Oops.
Yeah,
Okay.
drop the link in the at the very end of the chat. Should we do a giveaway and then we'll take a few more questions?
Yeah. I guess.
And so the first person to answer in the chat. So everybody get your keyboards ready.
Let me see. I don't want to make it too hard, but I have faith in you guys. So what is the actual piece of machinery that copies the DNA, that uses the primer site as its start site and makes copies of DNA?
Ooh, let's see. We've got
Ben got it.
ah,
So the thermocycler is the machine that does it, but it's actually the polymerase is the little, and I did say machine, so I was a little confusing. Sorry, guys. But DNA
right.
polymerase.
Ben, if you can send me your address
That's right, Stephanie. It is an
at
enzyme.
info at I'm going to drop the email address in the and I'll get a calendar out to you. There's
Um,
a few questions on the on the YouTube as well, so I'm going to jump over there real quick.
while you're doing that, I'm just going to go over this blast and NCBI
Okay.
real fast just for people. So if you just do a search for NCBI blast, these first two terms, this comes up. And so if you click on nucleotide blast, if you have a sequence, you can just put it here, copy paste it in. Let's see, do I have, I think I might have one I can do. Mm, sorry, I'm trying to pull up one real fast. Okay, here's one. Okay, so if I put one in, so it's separated it out, but that's okay. You don't have to do anything for that. And you don't really have to click anything here and blast it. And so what this does is it will compare it. Another option, if you're looking for a sequence, you can type in a species here. So and so this will pull up all the sequences associated with the lentinus. Oop, I must have hit a wrong button. Okay, so this was the blast that I just did. I put that sequence in and it compared it to everything. So it just shows you the first 100 similar sequences that pop up. So you have the name here. And then over here, it gives you a score. So the query cover shows how many of those bases match. So 98% did. Or how much of the query, let me go to the graphic summary. So how much, this is my query that I put in. This is the one it matched to. So it had a 98% linked similarity. And then 99.21% of the bases match. So I misspoke there. So if I click on this, it goes down and it shows me. So this was my query, the one that I put in, and this was the top match for it. So you can see mine is missing a base right here and right here that the one that it matched it to had. And then you can see all the other matches that it had. This one was a base difference. My query had a G, the one that it matched to had an A. So there are some differences here. And then I can click and look at the sequence more in depth. I can find out, more information about it. It has an accession number that's unique to it. I can figure out what paper it came from. So this is from a paper on gene family expansions and transcriptome signature and cover fungal adaptations to wood decay, which sounds interesting, and all the authors. And then I have the sequence here. So if you publish a paper, you have to deposit all of your sequences into NCBI, which is where these come from. Yeah.
Okay. I just wanted to mention too, before we take a few more questions for everyone, this class is recorded. And a lot of our online classes are recorded and put on our YouTube channel. So you can always kind of go back and pause, take notes, also get on some of these websites that Liz is demonstrating and try things out yourself. So you can always kind of go back and relearn and kind of catch up. And that link, the links on our YouTube, or if you received a confirmation email from us, the link is also there. So yeah, so I'm going to jump over to YouTube real quick. So we have Royce asks, I was wondering about how the taxonomic implications of collapsing anamorphs and telomorphs into the same species. Do we primarily look at DNA in those cases? Is it easier with metagenomics?
Okay, so that's a very good question. So what he's talking about is previously, before DNA, you have fungi that produce sexual morphologies and then asexual. And so before you had sequencing technology, those were generally described separately, so they would have two different names. So I can't remember which is sexual and which is asexual in terms of anamorph and telomorph. So sorry. It's not really used a lot. But then there was this huge conference in the early 2000s where all the big mycologists came together and decided that it was really dumb to have two names for the same fungus, and it was very confusing because it was. And so they decided to have one fungus, one name. And which caused a whole lot of issues. And there are still mycologists who are upset about it and will get into fighting matches about what name is the correct name. But yes, I think in general, DNA helped resolve those differences of whether they were the same species or not, because you do have different physical characteristics when you have asexual and sexual. It's a good question.
Right. Kylie asks, are there any examples of two fungi species that look identical but have only slight variations in its DNA that make one fungi poisonous and the other not?
Hmm. I can't think of one off the top of my head, but there was not poisonous, but could cause a disease. So I kind of referenced it. When I was in grad school, there was a woman, Allison Harrington, a graduate student who was in the lab I was in, and my PI, so my advisor, Betsy Arnold, who works on endophytes, they were trying to find fungi endophytes that were beneficial, and they found this one that was really beneficial. I think it helped with disease resistance and drought tolerance. And so they were trying to get it certified to be used in agriculture. But when they looked at the DNA, it was 100% similarity, so no base differences at all to a fungus that caused disease on the exact same host that this endophyte was beneficial to. I want to say it was in the genus Coniochida. And so to get it certified, they had to go through a whole process of sequencing other regions and doing morphological studies and describing it as a new species, and then to prove to the USDA that it was actually beneficial in a completely different species. So that's the only one I can think of. I can't think of one that's poisonous, though. Sorry.
So do you know if that particular fungi was ever certified to be used in agriculture?
I think so. I'm
Okay.
not sure though. I should follow up with her and ask. It was a really cool project.
Yeah. Yeah, there's so many cool things about endophytes. I love reading about them.
Yeah, they're fun.
Okay, so Cape asks, do you use P microspora for your plastic degradation project? If
Hmm.
not, can you share what species you are using and how are you approaching? Can you share how you set up your projects? Like
Um,
another talk.
yeah. So I can't share a lot because it's proprietary, which is something that's kind of new to me, in academia. That's one good thing about academia is You can share everything that you're doing, and you can talk about things openly. But that's something going to industry side, I've had to really kind of curve. So I can't tell you what species I'm using, but I can tell you, because it is in the literature and it's widely known, that a lot of the enzymes that fungi use to degrade recalcitrant plant materials like lignin, which is really hard to degrade, and fungi are pretty much the only organisms that can degrade it, can also degrade really recalcitrant plastics that take a long time to degrade. So I'll let you delve into that more, but you want to look for like white rotters and brown rotters and the enzymes that they produce.
Mhm. Yes. Let's see. Um, Ben had a good question about which we can get into, I think, after we talk about DNA,
Correct.
um, which is around academia as well. Um, but Keith asks, can environmental DNA samples be used to quantify the amount of different fungi in the sample, or is it mainly showing who is there, but not how much of each species?
So a very good question. Keith, I get the feeling that you know quite a bit already about sequencing. But no, so you can't really quantify DNA. And so with bacteria, which in my opinion are much easier to work with, but a lot less interesting, you can use, these environmental sequences, the reads that you get. So when you get all of these reads back, you end up with hundreds of thousands or more. You compare them, the sequences, the bases in them, and look at differences. So if I use a 95% sequence similarity, then sequences with more than 5% differences in their base pairs, I would consider different species. And so we don't really call them species though, because we can't prove that they're species because we're only looking at 250 base pairs. What we do is we call them operational taxonomic units, or OTUs. And so for bacteria, you have one species called an OTU, and you have all the reads that match that one that have 5% or less differences. So they're considered the same species. And so you can use like all the reads from that as kind of like a metric of abundance. But for fungi, you can't. And the reason for that is because the ribosomal DNA region in fungi, there's actually multiple copies in it in different taxonomic groups. So some taxonomic groups will have just one copy of this ribosomal DNA, and others will have hundreds or thousands. And so when you sequence that region, it doesn't just sequence one copy of it, it'll sequence multiple copies. So you don't know if you have 1,000 reads that match this species, you don't know, is it different individuals within that sample, or is it two individuals that just have a ton of copies? So you can't. A way around it is you can use qPCR, or you can measure, oh man, it's escaping me. I haven't used it before in a long time. It's or gas, no, I can't remember. I'm sorry. But you can measure like something else within that's in fungi, and you can quantify it, and it gives you like a rough estimate of how abundant the fungi are, but you can't look at species level differences.
Hmm.
So that is a shortcoming of environmental sequencing for fungi.
So Keith said ergustrial. Did I say that
Yes,
right?
thank you. Yes, I appreciate that. Yes, that's what you have to look at. You do know a lot about this.
Well, we're going to take one more question from Josh over on YouTube, and then we're going to switch gears. And Liz is going to tell us a little bit about her path from undergrad to getting her doctorate. And then we can take some questions about the academic path of mycology. So Josh asks, um, let's see, how would I best go about finding all the compounds in Gymnopolis luteifolius? Luteifolius? Is that done with HPLC testing or barcoding? I'm very new to this.
Okay, so you want to look at the metabolome. So what type of secondary metabolites are produced, I'm going to guess. So what type of chemicals are produced? So that isn't done with metabarcoding. I guess it could be if you know the specific regions you're looking for, but usually you don't. You know, usually if you're doing something like that, you're kind of mining fungi to see what types of chemicals are produced for either medicinal purposes or other purposes. So you would have to use HPLC and extract. First, take your fungus, extract all of the chemicals from it, and a method that uses a lot of chemicals. It's kind of hazardous to do, more so than extracting DNA. And then you would have to run it on HPLC. There might be other methods. I feel like I read something, maybe a slightly easier way, but it's not my area of expertise.
Yeah.
But you might want to look at metabolomic studies, that type of thing. And there's a postdoc now at UT called, oh crap. I'll post his name in the chat, or oh, you're on YouTube.
Yeah, you
Crap.
can send it. I can add it on the recording too.
He's at BFL. He does plant metabolomics, but it's
Okay.
a similar process for fungi too.
Yeah, that question's come up for me too recently, and I've reached out to a couple labs that do different panels for fungi and plants. But the challenge, we've been getting all this press about the Texas star mushroom.
Mm.
A lot of people want to know what's in it. Is it edible? You know, that's usually the first question people ask, which seems silly because it's got so many other interesting talking points. But
I feel like the texture would not be good, even if the
yeah,
flavor.
yeah, I'm like texturally, it's got little hairs on it. It's very leathery. I mean, I'm sure some chef somewhere could do wonders and
Oh yeah,
mean like animals eating it, but I can't seem to find any what did you call it? Metabolic,
metabolomic.
that's a tongue twister for me. Any of that type of research. So I thought it would be great to like spend a little bit of money and try to find out like what's in it. So I don't know. I may need your help digging and finding if it's actually been done before because I know this mushroom has been studied quite a bit considering its rarity. Um, and maybe when they did the Harvard study, like back in 2012, they also did that type of analysis. But yeah, if you don't kind of know what you're looking for, I know it's really hard to sort of do that type of analysis from what I've heard. You don't know what metabolites you're looking for.
Yeah, you kind of have to do kind of like this pull of everything and then look at it. So Brian Cedio he's a professor at UT. He looks at metabolomics of plants, and then Colin Morrison is a postdoc at BFL who's doing work on that too. So look at some of their papers and might be able to point you in the right direction.
Okay, great. All right. Well, let's switch gears. And so for people that aren't necessarily wanting to stick around for the latter part of this conversation, thank you for joining us tonight. It's been wonderful having everyone here. Lots of great questions. But yeah, so let's just kind of switch gears and I'd love to hear from you, Liz, like kind of on your path, like how you chose the academic path and some of your experiences, and then we can get into some questions.
Definitely. So I definitely did not start out my academic career looking at fungi. I was interested in plants. And I did my undergraduate in botany at Oregon State University in Corvallis, which if anyone who has been in Oregon knows, you kind of just fall into fungi very, very easily because they're everywhere. And so there were some really good mycologists up there who I took classes from. Joey Spatafora and Bruce McCune are two. Bruce McCune does more lichens, but I kind of fell into it with luck, and I got a job in an ectomycorrhizal lab. I was taking a mycology course, and there were two graduate students who were sitting right in front of me, and they were talking about how they needed an undergrad to do a bunch of really boring lab work. And so I kind of like inserted myself into their conversation and asked if I could please be that undergrad student. And so that was my first introduction to, kind of plant fungal relationships. And I really fell in love with it. I did some field work with them in Eastern Oregon, which was really cool, in the pine forest out there. That was a lab with the U.S. Forest Service looking at the effect of burns on ectomycorrhizal communities in these like clear-cut forests. And so I got to look at a lot of ectomycorrhizal colonized roots under a microscope, and it was really fascinating. So I started looking at grad schools. I was not sure that I wanted to do it at that time. I go to school for even longer. I'd been in the military before, so I was late to go to school, and I wasn't really finishing my bachelor's until I was 28. And so, I was ready just to not be in school anymore. But I ended up getting an NSF GRFP, which paid for my school. So that kind of convinced me that, it was worthwhile because I wouldn't have to pay for it. And so I was initially just going to get a master's, and I'd found an advisor, Betsy Arnold, at the University of Arizona, who was doing work on endophytes. So these are fungi that live in all plants that have been studied. They live in leaves and wood, in the roots, and they don't cause disease. They can be, beneficial, or they could just be hanging out doing nothing, kind of waiting for the right situation to emerge and find their niche. So I started out doing a master's, and I really liked the project that I was working on. I really liked what I was doing, so I switched to a PhD and spent 6 years there. Yeah.
Oh, great. Um, so Ben asks, what experiences do mycologists in academia look for in potential grad students?
So that's a good question. I guess it depends on what you're interested in doing. If having lab experience is always good, especially if you're planning to go into a more traditional type of lab that does kind of like knockouts and things like that. Having lab experience is good, having some familiarity with fungi. So I think you guys being part of CTMS and working with fungi, going out on walks, just like reading about fungi, really puts you ahead of a lot of people, especially where I was. But it really depends on the professor. Some are completely fine taking someone completely new with no experience, and some require experience. So it really depends on the professor and the area you're going to go into. And this is something you should have a conversation with them about. I think a lot of people are hesitant to talk to professors about what they want, but professors love talking about their research. And if you just read a few of their papers and just get them talking about the research, just ask them questions, they will talk forever about it, and you will endear yourself to them so much. Because usually when people talk about their research, you just have people's eyes glaze over. So having someone who shows interest in your research, it makes them feel good, and they will engage with you. And that's the best.
Great. Ben, do you have any follow-up questions? Maybe you want to take unmute and ask any follow-up or
It's cool if you don't.
okay.
Yeah, sure. Yeah, I'm a biochemist. I do a lot of lab work, but I don't have a lot of field work experience. Do you think like going on like mushroom walks and stuff helps with that?
So I guess it depends on like what you want to do. Like if you want to do ecology, then definitely going on walks, looking at fungi, would be helpful. Having lab experience is important. I think even, I'm a tried and true ecologist, but most of my time was spent in the lab. I would spend a couple of weeks every year, every season out in the field collecting samples. And then most of the rest of my time was spent in the lab processing those samples, and then analyzing the data and writing reports. So, not a lot of time is spent out in the field, definitely. So I think having that lab experience is useful. If you don't like ecology, you want to go into more, like taxonomy or things like that, then you just need lab experience.
Mhm.
Mm-hmm. Gotcha.
Okay.
Thanks. Yeah.
Most, like most, even in academia, like most work is done in the lab, not necessarily out in the field.
Yeah. Yeah. Which, being in the field is nice, definitely. You miss it when you're in the lab, and when you're in the field, you wish to be in the lab. But
Mhm.
I will say though, most degree programs out there, I don't know, it probably hasn't changed that much, but you usually will not be able to get a degree in mycology. Usually most mycology classes and degree paths are kind of subsumed within other degree paths. So, plant pathology. My PhD and master's is in plant pathology. And then you might end up in like EEB, so that's ecology and evolutionary biology. You could end up in microbiology. So usually you will not find a degree in mycology. If you do, though, let me know, because I would be interested to know if there is one.
Mhm. Since you've left the academic world for the private sector, have you noticed, um, just sort of peripherally, or maybe through just kind of keeping up with your academic pals and people in like some of the groups, do you feel like there's becoming more research opportunities or more studies with this sort of emergence and people's consciousness growing around fungi, like in the popular culture? Like, is that mirroring in academia at all?
Not really. I mean, you have certain areas of the country where you find more mycology occurring. So like West Coast, most definitely. Florida has some programs. You have Matt Smith at the University of Florida. There's some people up in like Minnesota, Michigan area, and then Northeast. But central US, especially Texas, you do not find a lot of mycology, even like UT. There are some people who like dabble with fungi for different projects, but there's no like true mycologist that works at UT,
Mhm.
which is surprising. But I will say, I mean, you can go the academic route, but there are quite a few, and I think ever increasing, I think more so in, kind of like the amateur, and I use that very broadly. These are not amateurs, just I guess non-degree holding mycologists are really increasing in number. So, I was mentioning it earlier, you have like David Lewis, you have, Alan Rockefeller, you have William Padilla Brown. You have all of these people who did not follow traditional academic routes, but are amazing mycologists and are doing probably more for the public field of mycology than many people who have degrees based in mycology. So you don't have to go the traditional route at all.
Yeah, one example, like thinking about, what you said about uh, the mycology being kind of intertwined in something that's not necessarily just mycology specific. When we last January, our guest speaker was Dr. Susie Dye at Texas A&M, and
Mm.
her lab is focused on cleaning up water. And I know she's doing a lot of different things, but working on removing uh, forever chemicals or PFAS from water. And so they're using fungi and working with, mycologists. Like, she's a trained chemist, but
Nice.
she's working with mycologists to degrade and break down these strongly bound chemicals. And so
Awesome.
it's not necessarily in the field of mycology, but chemistry. And I've seen some other things like that happening at UT, like even I know there's been some labs looking at like the enzymes that break down plastic as well at UT. I don't know if that research is still going on or.
I think so. I think we've connected with a few people at UT. Christine Hawks used to work at UT. She's a really awesome mycologist. She's now at North Carolina, and she is not like a traditional mycologist, but she's done all of these different studies of endophytes and ectomycorrhizal fungi in Texas, probably the most. But her degree is in environmental studies. And she has a PhD in just biology, so just general biology. So yeah, so there's many different ways to get to the same place, and not all of them go through academia. I have a lot of strong opinions about academia. There's a lot of issues in academia, and it's sometimes very inaccessible.
Mhm. Heather, do you want to come on off mute and ask your questions?
What
I can
degree?
ask them too.
Hello. So I'm just kind of getting started, and I'm almost 40, so I'm just, I'm working on a second degree or a second career rather. Anyway, I'm set to get an associate's in biology and associate's in general sciences in the summer of 2025 from ACC. And I've just kind of been trying to look at like what are the pathways and what are the ways to go for, a focus in mycology. And there seems to be a lot of different ways. And I'm not quite sure how or where or really what to look for. You know what I mean? Like, I'm not, it all seems a little overwhelming to me, the very many different ways that you can go with it. And so I just was wondering, maybe you had any advice on how to pin that down.
Yeah. So I guess, what you could do is look at papers. So you're working on your associates. Is the school you're going to, do they have access through the library to different like research papers?
Yes.
Awesome. Okay, so what I would do is just look up papers of subjects you're interested in. So if you're interested in like mycoremediation, so removing these forever chemicals from soil, if you're interested in like general ecology, if you're interested in taxonomy, just look up something. If you have like a specific species of fungi you're interested, you can look up papers on that. And read papers on them, look at the people, the authors that wrote them, and you can find other papers that they've written. If you have questions, reach out to them, ask them questions. Generally, they're good about answering. If they aren't, then that's, nothing to do with you. They're probably just really busy or maybe just horrible at correspondence. Unfortunately, people in academia are not very good at science communication, even though they should be. So yeah, just start reading about things that you're interested in and reaching out to people. It's not, a very definitive path, but that's the best method to kind of figure out what you're interested in, and people who are in that same field.
Thank you.
Yeah. And if they're in this area and you're near them, you can always reach out and ask if they have any openings in their lab. And you might have to start off volunteering. As I mentioned, people in academia are kind of poor most of the time. You have to work off grants, especially ecology labs. There's not a lot of money in that, not like engineering or some other more glamorous areas of academia. So you might have to start volunteering and then move into a paid gig.
Great. Thank you.
So Royce on YouTube asks, how does your academic experience compare to industry? Asking as someone trying to decide between industry and graduate school?
Yeah, so it's very different. So in academia, when your metric of success is how many grants you get and your publication and research history. So you are really trying to wring every bit of research out of a subject. And so if you have a very curious mind, it's awesome because you can do a study, and then from that study you come up with a million other things you want to look at. So like for my postdoc, my general area that was given to me in the job description was, we have this invasive grass, skinny grass, coming to South Texas, and we want to understand how it's changing the fungal community, which is very broad, So I was able to design projects looking at decomposition rates. So how have the decomposer communities changed? Have there been differences in carbon cycling, nitrogen cycling? Have there been changes in like these plant soil feedbacks that can occur? So if you have changes in the soil nutrients and plant community, that can lead to further changes, kind of like this positive feedback that reinforces this invasion. Has that occurred? Which it had, which was really interesting to find out. And then, in South Texas, they have a lot of grazing, and they do a lot of like clearing of these mesquite areas, which are really nutrient rich. So how does that impact invasion? Are these really detrimental? Does that harm like the natural native fungal community that's there and really allows this plant to come in? So you can design a million studies to look at this one area, and you can just like delve in deeper and deeper and deeper. In industry, you can't really do that. So you have your main goal and metric of success is a product, because you got to make money. So you can design really interesting studies, but you have this limitation of more so, is it helpful to what we're trying to market? But you have a lot more funding available in industry than you do in academia. So you're able to do some cooler things. Like I've been in this position at Hiro for a year, and all the equipment we've bought has been brand new. I did a PCR the other day, and usually in academia, when I did a PCR with the pipettes, which is how you move volumes around, this is a very specific example. In academia, everyone uses them. They're not really calibrated that often. And so when I did a PCR, I'd have to like account for extra volume that would just be lost due to like pipetting error. But we have these new pipettes that are very accurate, and I had set up the PCR accounting for this pipetting error, and I didn't need it. I ended up with like an extra volume of PCR master mix that I didn't need, that I had to throw away, which I felt really bad about because it was wasteful. But that's because I have this new equipment that's so accurate. So there's trade-offs, is what I'm trying to say. I will say, though, that the pay in academia is not very good. So if you go into grad school, you should not pay for it. If you are not offered a teaching assistantship or research assistantship, do not accept that position. You should not pay for grad school at all. You are not going to make a lot of money. I think in grad school, for the 6 years I was in grad school, I made about $16,000 a year, which was not much. And I had 2 kids, so we were very poor. But when I entered my postdoc, I thought it would be making a lot more money. It was more money, but it was not that much more. Postdocs, so I was looking around 2020. Everywhere I looked, postdocs made about $50,000. So there was a really awesome ectomycorrhizal lab in Boston that I really wanted to join, but they paid $50,000, which living in Boston, that's impossible. So I ended up not taking it. So you're not going to make a lot of money. In industry, you make a lot more money. So there's trade-offs, I guess. So you just have to kind of consider it, and you can always change, especially now. Like previously, I think if you were in industry, there was always this myth that you couldn't go back to academia. Once you left academia, you were out. But especially now, because it's so hard for people to stay in academia and they're losing so many people to industry, they really have to teach a little bit about industry and account for that. So I think you can go back to academia if you start out in industry or leave academia and go to industry. You can always find a place to return if you want to.
It's a good ender. I don't see any other questions. Anybody else have anything they want to unmute and ask before we say goodnight?
I can put my email if anyone has questions about academia versus industry. I'm happy to answer. So that's my email. I don't know if people on YouTube can see it, but
I'll drop it in the chat.
okay, feel free to message me if you have questions. I'm happy to help, or if it's questions regarding this presentation, clarification or whatever, I'm happy to do that as well.
Right. Well, thanks everyone for joining us, and especially thanks to Liz for such a great talk and making complicated scientific concepts very clear. And we hope to be doing more DNA or sending things up to Ohio and Indiana, where they have a machine going all the time, DNA sequencing, finding new species. So yeah, so we hope to see everyone at a future event. Check out our calendar, and everyone have a good night.
Thank you everyone for being here.